So we are going to be doing our presentation on the human microbiome and obesity. We are going to be doing parts 1 and 2 of this presentation. My particular portion of this presentation is on the current state of knowledge, links between the gut microbiome, host metabolism, and obesity. So the human gut is inhabited by a complex and metabolically active microbial ecosystem. That includes bacteria, bacteriophages, other viruses, yeast, fungi, protozoa, and archaea. It's been approximated that that there are about 100 trillion bacteria and more than 1,000 species of archaea. The microbiota plays a role in the induction, training, and function of the host immune system. When it's operating normally, the gut microbiota immune system alliance uses both the innate and the adaptive branches of the immune system to respond to pathogens and invaders. As the links between the gut microbiota and immunity become more clear, so too do the connections between microbiota and brain function. Studies that have examined microbiotic use and psychiatric effects in mice have found that there is an increased risk of depression and anxiety rising with consecutive use of that antibiotic. Diet is also known to influence brain function, and I will discuss that later as it affects the gut microbiome composition. In terms of the microbiome influencing the host metabolism, later in this presentation, we'll discuss in what ways the commensal bacteria can regulate or influence gut metabolism. Diet plays a large role in the gut metabolic profile of the host because what the host eats adds nutrient induced selective pressures on the bacteria inside. Bacteria that have evolved genes that can break down and metabolize specific nutrients are more likely to thrive in that environment that has those nutrients. Plant-based diets, plant-based diets and the use of prebiotics increase luminal fiber and complex carbohydrates. This environment selects for bacteria that have genes that code for enzymes that break down those complex carbohydrates. Animal-based diets that are rich in protein and fat and low in carbohydrates and fiber increase luminal bile acid content, and that selects for bacteria that can withstand those bile acids and also for bacteria that can metabolize those acids like bile acid hydrolases and sulfite reductase. Diets rich in fiber is a large influence on gut transit, which impacts fecal microbiome and metabolism. Without dietary fiber, it's been found that there is a shift in microbiome populations with an increase in bacteria that have mucin degrading enzymes. Desai et al. Suggests that this is due to an overall microbial preference towards fiber based substrates. Non nutritive sweeteners include aspartame, saccharin, and sucralose. They are used to help reduce people's sugar and calorie intake. However, it's been found in mice that use of these sweeteners hasten the development of glucose intolerance and increase weight gain in males. Saccharin has been found to increase the number of Bacteroidetes and decrease numbers of firmicutes. It's also been noted that the way that non-nutritive sweeteners affect microbial composition and metabolism in humans is largely determined by the bacteria that were already in place before use of these sweeteners. Bacteria in the gut play a role in the types and the amounts of hormones that are released, and in that way they can influence or regulate the host metabolism. Enterochromaffin cells are a type of enteroendocrine cells that are found along the lining of the intestine and the lumen. These cells are responsible for approximately 95% of serotonin in the body. These cells are sensitive to bacterial aromatic metabolites that are produced as bacteria digest the substrates available to them in the intestines. In turn, the serotonin, also known as the happy hormone, may influence the reward feeling that the host feels when eating a particular food. Glucagon like peptide 1 is released after eating and in response to nutrients like glucose. It also slows or inhibits gastric emptying and influences satiety and food intake. The gut microbiota can produce an enzyme called dipeptidyl peptidase 4, which influences levels of circulating GLP-1 and PYY, which I'll talk about in a moment. And that may create a feedback loop that can influence both metabolism and microbiota composition. Peptide tyrosine, PYY, like GLP-1, is highly expressed in the lower intestines. Increased amounts of PYY are associated with anorectic effects. So that's lowering of the appetite and reduced hunger. So as gut microbes exist and metabolize their food, they produce short-chain fatty acids, such as butyrate, that can increase the PYY gene expression. It's also been noticed that use of antibiotics increase PYY expression, but it's not yet known how and which bacterial species perform these alterations in that gene expression. Glucose-dependent insulinotropic peptide is a gastric inhibitory peptide. It's also known as an adipogenic peptide that promotes lipid uptake and adipocyte storage. GLP also influences populations of firmicutes and bacteroidetes in the gut, which in turn influence other hormones being produced. And finally, cholecystokinin is a hormone that's typically released in response to the intake of dietary protein and fat. It plays roles in appetite regulation, release of acids and enzymes during digestion. Less is known about this particular microbe and how microbes influence CCK, but studies are being done to investigate it further. So this is a graphic that basically just puts into context everything that I've been explaining. Starting at the top, we have diet that, as the arrow goes on, it's influencing which bacteria are being selected for. Those bacteria not only are producing those short chain fatty acids and secondary bile acids, they are influencing what hormones are being produced. There's the GLP-1, the PYY, the serotonin. Those hormones are influencing metabolism, and also the bacteria are directly influencing metabolism. So the link between gut microbiome and obesity has traditionally been very difficult to nail down due to the many facets and factors that play roles. We know that the diet strongly affects the diversity of microbes and other metabolites because of the selective pressures that specific substrates have. The richness and diversity of the gut microbiome do correlate with metabolic function, and an obesogenic microbe has been established, which has a high ratio of firmicutes to bacteroidetes. Studies have shown that overuse of antibiotics like vancomycin and ciprofloxacin have been linked to obesity and type 2 diabetes, but they also may have a role in downstream effects on metabolism. So interventions to shift gut microbiota may become a powerful tool in the future to help with metabolic disorders and obesity. Thank you.
Okay. I'm going to be talking about the challenges and opportunities currently and in the future for the microbiome and obesity types research. One standard that I found in a lot of these types of research, whether clinical or transitional, is they used the diet as the variable that they're going to be changing. They would originally check what your biome is, and then they would want to find out what would happen if they changed that diet over time. One that they use a lot that shows a lot of promise is the Mediterranean diet, which is the Med D diet. And that really has a focus on physical activity, remaining active as like the main basis of their pyramid. And then beyond that, focusing your meals on a day-to-day basis around fresh fruits and vegetables. And if you eat grains, you're going to have whole breads and rice. You're going to have whole grain varieties of these rather than a white bread like Western diets usually do. When it comes to drinking, that would be mostly water and in moderation wines. Meats would be a 2 to 3 times a week kind of thing. It's not every meal has to have a meat at the center. Like in America, that is a very prominent style of eating. Diets that are high in a change of oil. So in the Mediterranean diet, you're focused more on like an olive oil, but there are some studies being done on different seafood, sea creature rich oils like the calanus oil that can be used as a supplement, which comes from a population of plankton. Then there are specific things like the ketogenic diet or intermittent fasting, which have shown certain effects on your gut microbiota. But one thing that even with all of these, that is a challenge to be faced is in when you have people who would be the people you are choosing for your study who already have obesity or are overweight, they have a bias already towards maybe wanting to eat more high fat foods. And it would be hard to maintain that. You say, okay, this is what you're supposed to eat every single day. They're not going to be in there. There's no way that you can be watching a human every day, making sure that they are eating exactly what you're supposed to eat and only in the time period that you gave them to eat. If they could just easily diet with their current scheduling or with their current lives, maybe they wouldn't have been already in the situation that they are in. If it was easy, they would have already done it, I'm sure. And experimental models, they have chose a lot of mice and rats. There are in vitro culture studies, but a lot of the ones that I had found were on mice and rats. And they could mostly be broken down in a very, this is a very quick way of explaining it, but they do a initial biome testing, and then they would have groups split up, whether they do one type of diet or multiple types of diet, depending on what they are attempting. They will change that diet and they will check the blood and of course the weights. And once you change the diet over time, you will check those things again and again. And you will report on the changes of biome, which there's more abundance of a specific species or a certain genera of microbiota. And that would be how you would determine this. So this is going to be a lot of DNA sequencing. And you'll see here, once you have extracted that DNA, you're going to kind of go into two different directions on this. There's a more amplicon sequencing, which will be more of like a 16S ribosomal DNA sequencing that will give you an identity of what is living in your biome, in your gut biome, and then a more like general, a metagenomic sequencing, which is going to give you all the DNA. It's telling you what is being done in your biome, the different processes that are happening by all of these different microbiota. This will be, of course, done on a sequencer, which is in your top right. This is an iSeq 100. This is used. There are actually a large variety of these different sequencers that are used in the different studies I had seen. And then in the bottom right, I put a osmotic. It is a machine, a little pump that will give you a dose on timed intervals of a specific thing. In a study that I found that I thought was very interesting, they used this to give mice calamus oil. And I thought it was very interesting, and it was very efficient in actually helping them not only lose weight, but it changed the microbiota, even though they were on the same diet as other mice. So I will continue talking about that here. The types of informatics that I had found, you're going to be looking at a lot of different Shannon-Wiener index, alpha diversities, and abundance, relative abundance of specific bacteria. So I focused on that, and I wanted to just kind of show you the types of graphs that would be more relevant to this type of research. So on the bottom left, we have a relative abundance of genus for 4 different groups of mice that were in this experiment. And in the first, it's NCD, which means normal chow diet. HFD is high fat diet. And then we get to the ones that had the osmotic pump. And that's HFD plus cal, which is the calamus oil. And then HFD plus X, which is a exenatide, which is a drug used for diabetes type 2 and has some dietary effect as a treatment. So you can see it's color coded for which bacteria type, which genus of bacteria were more abundant in that collection, in that sample, when they were doing their fecal and blood samples. And you could see that for a lot of it, it was unassigned, but for the normal chow diet, there was a greater abundance in Allobaculum, whereas in very little in terms of Lactococcus, whereas in all of the high fat diets, there was a higher abundance of Lactococcus. And you can see the differences in the different high fat diets, with the only difference being at one point they had these osmotic pumps start giving them these different treatments. And then in the top right, you have an alpha diversity index. That's for Shannon-Wiener. And this is going to just tell you the higher the score, it's going to be a better diversity. And while it seems that the high fat diet has the highest diversity, it also, when you look at the abundance, it shows there is things that do better in that environment. And that would just tell you what is there. And then with your knowledge that you have prior to this research of what these specific bacteria are, you can use that information, the data given here, to make a discussion, find what is concluded from this information, from this data. And this is another one. This one's actually more specific. It's beyond the genus, and it's just another relative abundance. And they split it up by the different groups by color coded. So red is NCD, the not normal chow, and then blue is the high fat, and then the green has the calamus oil, and the HFD plus EX orange is the exenatide. And it just shows the difference. Like the alobaculum is very, very abundant in the normal chow. And then you can kind of see where the green is much larger for lactococcus and lactobacillus. So this kind of information would just tell you what is there, and then you can use that to determine how those bacteria are affecting your gut microbiome. And this is my references. Thank you.
Thank you.